Your qPCR result looks fine. Clean curves, strong signal, everything worked. But that doesn't mean it's correct. qPCR doesn't tell you what you amplified. It only tells you that something amplified. And PCR is very enthusiastic. Problem 1, contamination. Even your blank shows a signal. Without a no-template control, you don't know if your result is biology or your pipette. Problem 2, measuring DNA when you think you're measuring expression. The -RT control tells you whether your signal comes from RNA or leftovers. Problem 3, differences that look biological but aren't. Internal or spike-in controls show whether samples are truly different or just handled differently. Problem 4, silent failure, no amplification, flat curves, no signal at all. Is the lack of signal real, or is it just a technical failure? Is the gene absent, or did the reaction fail? Without a positive control, you can't tell. Problem 5, variability. Replicates tell you whether a result is reproducible or just lucky. 1 well is an opinion. 3 are data. Controls don't complicate qPCR, they make it honest. They don't guarantee perfect experiments. They prevent confident mistakes. In our self-paced qPCR training, we show how to choose, interpret, and trust these controls. So your results don't just look good, they mean something.